Woolly mammoth. The woolly mammoth was a massive, fur-covered elephant species that lived across the frozen tundras of the northern hemisphere during the Ice Age. It went extinct roughly 4,000 years ago, likely due to a combination of climate change and human hunting. Modern scientists believe the mammoth is one of the most realistic candidates for de-extinction. Its DNA has been recovered from frozen carcasses preserved in Siberian permafrost. In some cases, soft tissue, blood, and hair have been found remarkably intact. Researchers are not attempting to clone a mammoth directly. Instead, they are editing the genes of modern Asian elephants to include mammoth traits, such as thick fur, cold-resistant fat, and hemoglobin adapted to low temperatures. The goal is to create a hybrid capable of surviving Arctic conditions. Some scientists argue that reintroducing mammoth-like animals could help restore grassland ecosystems and slow permafrost melting. Critics warn of ethical concerns and unpredictable ecological consequences. Despite debate, genetic work on mammoth revival is already underway. Tasmanian tiger. The Tasmanian tiger, also known as the thylacine, was a carnivorous marsupial native to Australia and Tasmania. It resembled a dog with tiger-like stripes and was the largest marsupial predator of modern times. The last known individual died in captivity in 1936. Thylacines were hunted extensively due to fears they threatened livestock. Their extinction occurred recently enough that high-quality DNA samples remain preserved in museum specimens. Scientists have successfully sequenced the full thylacine genome using century-old tissue. Modern de-extinction efforts aim to reconstruct the thylacine genome using closely related marsupials as genetic templates. Unlike dinosaurs, thylacines share many traits with living species, making genetic reconstruction more feasible. Some researchers believe the thylacine could be reintroduced to help control invasive species in Australia. Others caution that the ecosystem it once occupied has changed dramatically, raising questions about its survival in the modern world. Dodo. The dodo was a flightless bird native to the island of Mauritius in the Indian Ocean. It evolved without natural predators and became extinct in the late 1600s after humans introduced hunting and invasive species. Despite its extinction centuries ago, usable DNA has been extracted from dodo bones and preserved remains. Scientists have identified the dodo's closest living relative as the Nicobar pigeon, which provides a genetic reference for reconstruction. Rather than cloning a dodo, researchers aim to modify the DNA of living pigeons to express dodo-like characteristics. This process involves selective gene editing to recreate the bird's size, shape, and behavior. De-extinction proponents argue that restoring the dodo could help repair damaged island ecosystems, as the bird once played a role in seed dispersal. However, Mauritius has changed significantly since the dodo's extinction, raising questions about habitat suitability. Saber-toothed cat. Saber-toothed cats, including Smilodon, were powerful predators that lived during the Ice Age. They are best known for their long, curved canine teeth and muscular build. These cats disappeared around 10,000 years ago, possibly due to climate shifts and the loss of large prey. DNA recovery from saber-toothed cats is more difficult from frozen mammoths, but scientists have extracted partial genetic material from fossilized remains. Advances in genome reconstruction may allow missing DNA segments to be filled using related big cats, such as lions and tigers. Unlike mammoths, saber-toothed cats would require large territories and abundant prey, making reintroduction challenging. Their hunting strategies were highly specialized, and modern ecosystems may not support them. Despite these challenges, saber-toothed cats remain a subject of de-extinction research, offering insight into predator evolution and Ice Age ecosystems rather than immediate revival plans. Passenger pigeon. Once numbering in the billions, the passenger pigeon was one of the most abundant birds in North America. Massive flocks darkened the sky for hours. Within a few decades of industrial hunting and habitat destruction, the species vanished entirely. The last passenger pigeon died in 1914. High-quality DNA samples have been preserved in museum collections. Scientists have already sequenced much of the passenger pigeon genome and identified differences between it and its closest living relative, the band-tailed pigeon. The plan involves editing the genome of modern pigeons to recreate passenger pigeon traits, including flocking behavior and reproductive patterns. Unlike single animal revival, restoring passenger pigeons would require large populations to function ecologically. Researchers see this project as a test case for whether humans can undo large-scale ecological damage caused by overexploitation. Aurochs. The aurochs was a massive wild cattle species that once roamed Europe, Asia, and North Africa. It is the ancestor of modern domestic cattle and went extinct in the 17th century due to hunting and habitat loss. Unlike many extinct animals, the aurochs left behind living descendants. Some modern cattle breeds still carry genetic traits of their wild ancestors. Scientists are using selective breeding and genetic editing to recreate aurochs-like cattle in a process known as backbreeding. Projects in Europe have already produced large, aggressive cattle resembling historical descriptions of aurochs. These animals are being introduced into nature reserves to restore grazing patterns that maintain open landscapes. Although these animals are not perfect genetic replicas, they function similarly within ecosystems, blurring the line between revival and reconstruction. Pyrenean Ibex. The Pyrenean Ibex was a wild goat native to the Pyrenees Mountains. It officially went extinct in 2000, making it the first species to be successfully cloned after extinction. Scientists used preserved skin cells to clone the animal, resulting in a live birth in 2003. However, the clone died minutes later due to lung defects. Despite the failure, the experiment proved that de-extinction is technically possible. Improved cloning techniques and genetic screening may allow future attempts to succeed. The Pyrenean Ibex remains a landmark case demonstrating both the promise and limitations of de-extinction science. Moa. Moa were giant, flightless birds native to New Zealand, some standing over 3 meters tall. They were hunted to extinction by early human settlers roughly 600 years ago. DNA extracted from moa bones is well preserved due to New Zealand's climate. Scientists have sequenced significant portions of the genome and identified genetic similarities with modern birds such as emus and tinamous. Recreating a moa would require using a living bird surrogate, presenting significant biological challenges. Ethical concerns also arise due to the bird's size and habitat requirements. While full revival remains distant, moa DNA research provides valuable insight into avian evolution and human-driven extinctions. Woolly rhinoceros. The woolly rhinoceros lived alongside mammoths during the Ice Age, adapted to cold environments with thick fur and fat layers. It went extinct around 10,000 years ago. Frozen remains discovered in Siberia have preserved fragments of DNA, though not as complete as mammoth samples. Scientists believe similar gene editing techniques could be applied using modern rhino species. However, rhinos have long gestation periods and low reproductive rates, making de-extinction efforts slow and complex. Conservationists argue that protecting living rhino species should take priority. Quagga. The quagga was a subspecies of zebra native to South Africa, distinguished by partial striping on its front half and a solid-colored rear. It was hunted to extinction in the late 1800s. Because quaggas were closely related to modern plains zebras, scientists have used selective breeding to recreate quaggas that closely resemble the extinct subspecies.